Hello everyone, and welcome to our channel, MooTechie. A special welcome to all the dairy technologists and students joining us today. We're diving into the world of food microbiology, focusing on the chemical changes caused by microorganisms and the sources of contamination that lead to food spoilage. Moving on, we explore how different chemical changes are possible in foods. This is because a great variety of organic compounds are present, and numerous kinds of microorganisms that can decompose them may grow in the food. These changes are broadly categorized into changes in nitrogenous organic compounds and changes in non-nitrogenous organic compounds. Now, let's look at changes in nitrogenous organic compounds. Most of the nitrogen in foods is in the form of proteins. Proteins are hydrolyzed into polypeptides, simpler peptides, or amino acids before they can serve as nitrogenous food for most organisms. Proteinases catalyze the hydrolysis of proteins to peptides, which can give a bitter taste to foods. Peptidases catalyze the hydrolysis of polypeptides to simpler peptides, and finally to amino acids. Anaerobic decomposition of proteins, peptides, or amino acids results in the production of obnoxious odors, a process called putrefaction. Next, we delve deeper into putrefaction. It results in foul-smelling, sulfur-containing products such as hydrogen sulfide, methyl and ethyl sulfides, and mercaptans, plus ammonia, amines like histamine, tyramine, putrescine, and cadaverine, phares, as well as indole, scatole, and fatty acids. When microorganisms act on amino acids, they may deaminate them, decarboxylate them, or both. For example, Escherichia coli produces glyoxylic acid, acetic acid, and ammonia from glycine. Pseudomonas produces methylamine and carbon dioxide from alanine. Clostridia produces acetic acid, ammonia, and methane from alanine. These three organisms can produce alpha-keto acid, ammonia and carbon dioxide, acetic acid, ammonia and carbon dioxide, or propionic acid, acetic acid, ammonia and carbon dioxide respectively. Following this, we examine other nitrogenous compounds. Desulfatomaculum nigrificans, an obligate anaerobe, can reduce sulfate to sulfide and produces hydrogen sulfide from cysteine. Other compounds include amides, imides, and urea, from which ammonia is the principal product. Guanidine and creatine yield urea and ammonia. Amines, purines, and pyrimidines may yield ammonia, carbon dioxide, and organic acids. Now, let's shift to changes in non-nitrogenous organic compounds. The main non-nitrogenous foods for microorganisms, used primarily to obtain energy but possibly serving as a carbon source, include carbohydrates, organic acids, aldehydes and ketones, alcohols, glycosides, cyclic compounds, and lipids. Starting with carbohydrates, they act as an energy source for microorganisms. Complex di, tri, or polysaccharides are usually hydrolyzed to simple sugars before utilization. A monosaccharide like glucose, when oxidized aerobically, would be broken down to carbon dioxide and water. Under anaerobic conditions, glucose can decompose in several ways. First, through an alcoholic fermentation by yeasts, producing ethanol and carbon dioxide. Second, a simple lactic fermentation by homofermentative lactic acid bacteria. Third, a mixed lactic fermentation by heterofermentative lactic acid bacteria, producing lactic and acetic acids, ethanol, glycerol, and carbon dioxide. Fourth, the coliform type of fermentation by bacteria like E. coli yeasts, yielding lactic, acetic, and formic acids, ethanol, carbon dioxide, and hydrogen. Fifth, the propionic acid fermentation by propionic bacteria. And sixth, butyric butyl isopropyl fermentations that yield lactic acid, acetic acid, and other products. Now, on to organic acids. Organic acids, often occurring in foods as salts, are oxidized by microorganisms to carbonates, making the medium alkaline. Aerobically, they may be completely oxidized to carbon dioxide and water. Saturated fatty acids or ketonic derivatives are degraded to acetic acid. Other compounds like alcohols are usually oxidized to corresponding organic acids, for example ethanol to acetic acid and acetaldehyde to acetic acid. Next, we discuss lipids. Fats are hydrolyzed to glycerol and fatty acids by the enzyme lipase. Phospholipids may be degraded to their constituent phosphate, glycerol, fatty acids, and nitrogenous base such as choline. For pectic substances, protopectin in plants is converted to pectin. Pectin is a water-soluble polymer of galacturonic acids. Pectin esterase causes hydrolysis of the methyl ester linkage of pectin to yield pectin acid and methanol. Moving on, we now turn to the sources of microorganisms causing food spoilage. Primary sources are where viable organisms are found in a wide range of habitats, from polar regions to hot springs, from acidic to alkaline conditions, and in the presence or absence of oxygen. They are also found on the surfaces of plants and animals. This normal flora can affect the original quality of raw materials, the type of contamination during processing, and lead to food spoilage or illness. Therefore, the normal flora from food, soil, water, or the environment plays a major role in microbial spoilage. Now let's talk about microorganisms in the air. Air serves as a means of dispersal rather than growth for microorganisms. It contains many gram-positive rods and cocci, including flora from animal or human sources or water. Common bacteria in air include Micrococcus, Corynebacterium, Bacillus, and Streptomyces. Common fungi include spores of Penicillium, Aspergillus, Fusarium, and Cladosporium. Coughing and sneezing generate aerosols that may carry viable microorganisms. Next, we explore microorganisms in soil. Soil has diverse microflora, including bacteria, protozoa, fungi, and algae, so food should be protected from dirt to reduce spoilage. Soil is a reservoir of resistant structures like endospores of Bacillus and Clostridium, and chlamydospores and sclerotia of many fungi that can easily spoil foods. For water wicks, fresh water from rivers and lakes has microflora from terrestrial animal and plant sources. It can act as a vehicle for bacteria, protozoa, viruses, etc., leading to spoilage if used for washing foods. Now, on to microorganisms on plants. Plant surfaces have natural microflora called phylloplane flora on leaves and rhizoplane flora on roots. This includes fungi like Cladosporium, Penicillium, and Aspergillus, yeasts like Sporobolomyces and Bullera, and bacteria like Erwinia, Pseudomonas, Xanthomonas, Lactobacilli, Streptococci, and Leuconostoc. Erwinia can cause blackleg disease of potato and soft rot during storage. Fungi like Botrytis cinerea may infect strawberries. Cereal grains can be infected by Cladosporium, Alternaria, Chitomium, Helminthosporium, Penicillium, and Aspergillus. For example, the surface of a well-washed tomato contains 400 to 700 microorganisms per square centimeter. The outer tissue of unwashed cabbage can contain 1 million to 2 million microorganisms. Following this, we examine microorganisms from animals. The intestinal tract of animals has many microorganisms that, if shed into water bodies, can cause food spoilage when used for washing. The skin also contains microorganisms like Staphylococcus and Corynebacterium that can contaminate food. Feathers and feet of poultry carry heavy contamination. Salmonellosis associated with eggs has been reduced due to pasteurization of egg products. Meat from slaughterhouses is not frequently associated with human salmonellosis. Utensils used to store harvested fruits and vegetables are contaminated with surface organisms, which can lead to contamination of other products placed in them later. Finally, food handlers themselves are a source. Microorganisms on hands and outer garments of handlers can contribute to the microflora of food. The nasal cavity and mouth are also important sources of microorganisms. Thank you for joining this session on food microbiology. We've covered the key chemical changes caused by microorganisms in foods, including the breakdown of nitrogenous and non-nitrogenous compounds, and the various sources of contamination from air, soil, water, plants, animals, utensils, and handlers. Understanding these processes is essential for preventing spoilage and ensuring food safety in dairy and related industries. Thank you. See you in the next class.